Here are the list of the members for this group. First, we got Nur Amalin, Muhammad Sofi, and Nurfahana Akila. MRSA is a major human pathogen identified as a serious threat by WHO and CDC due to its resistance to multiple drugs and ability to cause severe and sometimes fatal disease and infections. One key of its resistance is biofilm formation, which shield the bacteria from antibiotic immune responses. MRSA's biofilm is composed of self-produced substances that protect it from environmental stress. The bacteria regulates their virulence through systems like accessory gene regulator and CRS, which control the expression of the various adhesion and virulence proteins. Recent treatment strategies focus on disrupting these virulence factors rather than killing it outright. Citral, a compound which found in citrus plants, is known for its antimicrobial properties and is generally recognized as safe by FDA. This study explores citral's ability to inhibit MRSA biofilm formation and its mode of action using proteomic analysis. So, the objective of this research are to investigate the citral effect of biofilm and growth of MRSA, and also to investigate the effect of citral on cellular proteome of MRSA. So, the hypothesis that can be made for this research is the citral can inhibit biofilm formation in MRSA by targeting specific molecular mechanism, which can be identified by using proteomic analysis.
The first method that was used in this article is phytochemical preparation. In this method, methanol was used as solvent control in all the assay performed. The second method is anti-biofilm assay. This method was determination of BIC, which is Bayesian information criteria. This BIC of citral was determined by performing anti-biofilm assay in 24-well MTP. The third method is image analysis, spot picking, and also in gel digestion. Gel image were scanned using a densitometric image scanner with the resolution of 300 dpi and captured through LabScan 6.0 software. Further, image analysis was performed through image master 2D platinum 7 differentially regulate protein spot with N2 fold change was selected and excised from gel for further identification. The first result that we can get from this article is the effect of citral on biofilm and growth of MRSA. The first one is result of MTP assay showed that citral inhibit a dose-dependent antibiofilm activity with maximum biofilm inhibition of 74% at 100 microgram per ml without affecting the growth. Second, it growth and alamar blue assay. The second one is the effect of citral on cellular proteome of MRSA. Resulting gels were stained with colloidal Comasi brilliant blue (CBB) and many protein spots with a differential expression were observed. Second, 2D GE image analysis were upregulated and downregulated protein spots are encircled with green and red color respectively. In addition, string and gene ontology analysis predict that most of the upregulated proteins are enzymes involved in metabolic process, cellular process, regulation of biological process, and also respond to stimulus, and down-regulated proteins are involved in cell adhesion, hemolysis, capsular polysaccharide biosynthesis, and also phytogenesis. The third result that we get from this article is validation of key result of proteomic analysis by in vitro assay and also qPCR analysis. The first one is qualitative assessment of the slime production. Second, staphyloxanthin biosynthesis and oxidant susceptibility. As staphyloxanthin is the antioxidant pigment, inhibition of the pigment has reduced the reactive oxygen species, which is ROS, scavenging ability of MRSA. This was confirmed by reduction in survival of MRSA in H2O2 buffer open citral treatment.
Conclusion, this study found that citral significantly inhibit MRSA biofilm formation at 100 microgram per milliliter without affecting growth or metabolism. Proteomic and transcriptional analysis revealed that citral upregulates the transcriptional repressor COY and protein involved in protein synthesis and metabolism while downregulating virulence factors such as hemolysis, lipase, and mycotoxins and staphyloxanthin. Given citrate ability to target multiple virulence pathway in MRSA without hindering growth, it presents a promising therapeutic option for combating biofilm-associated infection. Its mechanism of action involving the regulation of a broad range of proteins suggests a lower likelihood of resistant development, making it a valuable candidate for further clinical investigation. Can any of the results be interpreted differently? Yes, the study emphasized that citrate functions mainly as an antibiofilm agent rather than antibacterial one. This distinction is important because antibiofilm agents aim to reduce virulence rate without necessarily killing the bacteria, thereby lowering the risk of developing resistance. However, the fact that citrate does not exhibit antibacterial properties at its biofilm inhibitory concentration (BIC) can be seen as limitation. This viewpoint highlights that while citrate effectively prevents biofilm formation, it does not eliminate MRSA bacteria, which could still pose a threat. Secondly, as the study suggests that citrate could be promising candidate for developing new treatment against MRSA infection, but critics might argue about the practical application of citrate in real-world settings. Question could be raised about the stability, delivery method, and possible side effect of citrate in clinical environment. Moreover, the potential of resistant development even against antibiofilm agents could be a point of contention. Limitation of research. First, specific bacterial strain. The study focused solely on one strain of MRSA. This limits the generalizability of the finding, as the result may vary with different MRSA strain or other bacterial species. Second, potential resistance. Although citrate was shown to inhibit biofilm formation, there is a possibility that MRSA or other bacteria could develop resistance over time. The study did not explore the long-term effect or potential for resistant development. Lastly, in vitro condition, the experiments were conducted under controlled laboratory condition, which may not accurately reflect a real-world scenario. Factors such as human immune response and the complex environment of the human body are not accounted for.